Hello students, good afternoon. So today we will continue about the chapter number 4 from the subject biology, that is the molecular basis of the inheritance. Yesterday we will completed about the Griffith experiment to prove the genetic material is DNA. But the Griffith failed to explain which one is the transforming principle due to which the R strain bacteria get transformed into the S strain bacteria. In short, again I will repeat it again. So the Griffith performed the experiment by using the Streptococcus pneumoniae type of bacteria. There are two strains as S strain and R strain bacteria. The S strain bacteria are the pathogenic type of bacteria, while the R strain bacteria are the non-pathogenic type of bacteria. By using the experiment on the mice, here in the mice the Griffith injected S strain bacteria, and the result, all the mice get died by causing the disease pneumonia. While in second step the Griffith added R strain bacteria into the mice and observed that all mice remains survived in live condition because the R strain bacteria are the non-pathogenic type of bacteria. In third step the Griffith added heat killed S strain bacteria into the mice and observed that the mice again remains survived. So as if the S strain bacteria are pathogenic, but due to the heat killing, here it becomes non-pathogenic, so the mice cannot cause by any disease, pneumonia, and so the mice remains survived. And in fourth step, the Griffith added R strain and the heat killed S strain bacteria into the mice, and the result somewhat the puzzling. Here, as if separately R strain bacteria added, the mice remains live condition. As if separately heat killed S strain bacteria injected into the mice, again it remains live or the survived without causing any disease. But as if both get added, both get injected at the same time in the mice, the result somewhat the puzzling one, that all mice get died by causing the disease pneumonia. And so later on the Griffith, which isolate the material, the extract from the blood of the mice, and through the extract he isolates some unique material, some unique substance due to which the R strain bacteria get converted into the S strain bacteria. The Griffith observed that here in the mice the S strain bacteria are present, which are the live bacteria, which becomes live. Ultimately the R strain bacteria get converted into the S strain due to the some material, and the Griffith only denote as it the transforming principle. And so later on, again to explain what is the transforming principle, the Avery and their associates, that means the cytologist Avery, then the McCarthy and the MacLeod's performed the further experiment, and it turned as the Avery, McCarthy and MacLeod's experiment, or in short it may be termed as the Avery experiment. The Avery experiment here used the Streptococcus pneumoniae bacteria, particularly the S strain bacteria. The S strain bacteria through which their extract, the Avery purify DNA, RNA, proteins and some other substances may be like carbohydrates, etc. So from the blood sample of the S strain bacteria, the aviary which isolates separately as DNA, RNA, proteins, carbohydrates, etc. And then later on, each substance, isolated substance, it get mixed with the R strain and the heat killed S strain bacteria. That means there may be the different steps. Suppose in a test tube, the aviary, suppose here the R strain bacteria. In the R strain bacteria, here the aviary added DNA separately. So suppose here the R strain bacteria plus DNA. Then again in another test tube, R strain bacteria and RNA. In the R strain bacteria, the RNA get added. Again separately in R strain bacteria, the proteins get added, like this. So here the aviary perform the experiment by using separately, suppose the R strain bacteria, and in which the different substances get added. Suppose plus DNA, R strain, that means the rough strain bacteria, and in which suppose here added RNA, R strain bacteria plus proteins, like this. So the aviary added separately the different isolated material or the compounds or the substances in the R strain bacteria, and then observe that as if in the R strain bacteria the DNA get added, and later on observe that the R strain bacteria get converted into the S strain bacteria. In second step, the R strain bacterium in which the RNA get added, then the result it observed that the R strain bacteria remains as it is. There is no any transformation, there is no any conversion. Maybe in third step, the aviary added proteins in the R strain bacterium. The result, the R strain bacterium remains as it is. So according to the aviary experiment, or the aviary carrying the different steps, and finally concluded that only the DNA is the substance or the material which converts the R strain bacterium into the S strain bacterium. That means the non-pathogenic bacterium get transformed into the S strain bacterium. And so here the aviary and their associates concluded that the DNA must be the genetic material. So here only the DNA is the genetic material. Which transformed R strain bacterium into the S strain bacterium. Again the Avery, for the conclusion, perform the experiment in further steps, like here the R strain bacteria and the proteins get added into the R strain bacteria. At the same time, the Avery added proteases. Proteases, that means the enzyme which lysis, which broken down the the polypeptide chain of the protein. That means it is a macromolecule, somewhat the complex substance, much elongated strand of the protein, and the elongated strand of the protein which can broken down in presence of enzyme proteases. And then the result observed that the R strain bacteria, it remains as it is. That means there is no any transformation. Again, in another step, the Avery performed that the R strain bacteria which added with the RNA, at the same time the enzyme RNase. So here the RNase, again the RNase enzyme which helps to lysis the polynucleotide chain of RNA. That means the RNA molecule can broken down in presence of RNase, and at the here the result, the R strain bacteria remains as it is. Similarly, the Avery also added here, this one the step, the R strain bacteria in which the DNA get added and the enzyme DNase added at the same time. The function or the role of the DNase, the enzyme which lyses the DNA molecule. That means the DNA molecule can broken down, and at that time, as if the lysis of the DNA get achieved in presence of enzyme DNase, so at that time the the R strain bacterium, it remains as it is. There is no transformation. Again I will repeat here, the Avery performed the experiment by isolating the different substances from the extract of the S strain bacteria, and each substance gets separately added in the R strain bacterium. As if in the R strain bacterium the DNA get added, it get converted into the S strain bacterium. That means the transformation get achieved here. But in the same experiment, as if the DNAs get added, the DNAs which lies or broken down the DNA, that means there is no effect of DNA get achieved in this step, and so the R strain bacteria remains as it is. There is no transformation get achieved, and so here the two steps are much important again. The R strain bacterium in which as if the DNA get added, the transformation get achieved, that means the R strain bacterium get converted into S strain bacterium. But as if in the same experiment the DNAs get added, then there is no transformation. The R strain bacterium remains as it is. And so finally again the Avery concluded that DNA is only the genetic material. what is the DNA according to the Griffith experiment and according to the evidence of the Avery experiment? The DNA is nothing but the transforming principle. The transforming principle, again here we can observe that in the Griffith experiment under the fourth step, here the fourth step, the fourth experiment, the R strain bacterium get converted into the S strain bacterium due to the transforming principle, and the transforming principle is nothing but the DNA and only the DNA, and that is the Avery experiment. Here the question may be asked for 3 marks, explain the Avery experiment, or maybe here also the weightage carried from the Griffith experiment also may be for 3 or 5 marks. So we have to cite here maybe under the Griffith experiment or maybe under the Avery experiment, both experiment which worked on to explain what is the genetic material. But the Griffith completely fails to explain what is the transforming principle, and the Avery experiment finally concluded that here the DNA must be the genetic material. So both experiments separately carries, the question may be asked 3 to 5 marks. What is the Griffith experiment, or explain the Griffith experiment, as well as maybe what is the Avery experiment, or explain the Avery experiment with the help of the different steps under this experiment. So in this way, in your textbook, the 2 evidences or the experiment get included to explain the genetic material as a DNA, and again the third one also get included, that is the Hershey and the Chase experiment. I will only introduce in short here, and then in next lecture I will continue in detail about the Hershey and Chase experiment. The Hershey and Chase experiment, so Hershey and Chase, that is the cytologist, which work again to prove the genetic material must be the DNA by using the bacteriophages. The bacteriophage virus. The bacteriophage virus, particularly the bacteriophage virus, I will only give about the short information, and then in next lecture I will explain in detail. So the bacteriophage virus, which is somewhat the tadpole-like structure, like this, having the head, the connecting link, and the base, that is the tail. So the head piece and tail piece, and at the tail here, which at the time of the infection, the tail piece which incorporate in the body of particularly the bacteria, which infect particularly the bacteria, and so it termed as the bacteriophage virus, in which inside it the genetic material is present, that is the DNA. The bacteriophage virus composed by only the 2 components. So here the coat, their coating, which one is the proteinous coat, termed as the capsid, and capsid composed by the protein, and inside the capsid here the genetic material is present, and the genetic material, which one is the DNA. The bacteriophage virus, the Hershey and Chase, which use the bacteriophage virus, and in the bacteriophage virus, the Hershey and Chase added the isotope of, by using the isotopes, the isotope of sulfur and isotope of the phosphorus. So I will continue in further lecture details about this experiment, the Hershey and Chase experiments.